Swayam Prabha. Digital India. Educated India.
Welcome back to my industrial biotechnology course. Now I was discussing about the brewing industry. Then in the last class I tried to share with you that what are the different type of beers that are usually available in the market, and what are the major steps are involved in the beer making industry, and how the hops plays important role to give the specific taste and flavor to the beer. And also I try to discuss the process flow diagram of the whole process to give you the overall idea on the beer making industry. Now in this lecture I shall continue this beer and go in details that what are the how these individual steps of operations, how they operated, and also we will give the detailing of the process that we also will work out. So we start with malting process, because this is the first step of this process, and I told you this is produced from the barley grains, and barley grains looks like this. I told you this is something similar to the wheat grains, so it looks like that. The quality of the barley grains that plays very important for the quality, that influence the quality of the beer. So in India that we are Kingfisher beer largely marketed not only India but also in the foreign countries. Now malting of barley grains is a malt is an enzymatic product prepared usually from the selected barley. The manufacture of malt commonly carried out by malting process, which consists of the following step: selection or screening of the barley grains, and then steeping process, what we call soaking process, germination process, what we call sprouting process, and drying under the careful regulation conditions. So the four major steps are involved in the malting process. Now it can be explained like this: the steeping process, your barley grains in presence of water. Initially the water contents of the barley grains is 12%. Then when you soak in the water, this barley grain in water, then it increases to about 45%. And then these barley grains we keep it. I told you that we keep it under aerobic conditions and under humidified condition, so that the germination take place. So what we call the enzyme induction and cell wall digestion that will take place, and we get the green malt, and then we dry this, and then we have the dry, and so that the moisture content comes to 4%. We make it powder, and we use as a malted food, or this is used as the raw materials for the beer making industries. Now selection of barley grains, that we have this grains are large or uniform size. This is very important, and those which has yellow color and when matured and high germination power should contain small amount of harsh and bitter substances in the husk, and should have the ability to produce high amount of enzyme during the germination process. Now steeping process, it looks like this. You soak in the water. You can see how it is there. It is a process of soaking barley grains so that it may take up sufficient moisture to quicken the living cells of the embryo and start the process of enzyme production and germination of the which proceed the breakdown of the cell wall and hydrolysis of the stored food. This is done in the aerobic conditions. After the steeping process, The final desired is the moisture concentration of the barley grains should be 45 to 47%. pH, higher pH is maintained, and steeping period usually for 2 days, and temperature we maintain 10 to 15 degrees centigrade. Now this is the barley grains, how it looks. You can see that how this we have pericarp, then endosperm, the germs that we have. Actually here the germination take place. You can see that here in this germination here ascospore formation is there. When this germination take place 3/4 as length of the kernel, then and only then you stop the germination process. How you stop the germination process? You just dry it, so the germination will be stopped. Now this is the sprouted barley grains. You can see how the shoot formation take place in the barley grains during the germination process. The germination enzymes are activated when the temperature, moisture and aerobic conditions are satisfactory for the germination of the seed. Germination generally required 5 to 7 days. The temperature of the grain maintains 60 degrees Fahrenheit to 70 degrees Fahrenheit by controlling the temperature of the incoming air. The process is complete when the ascospore has grown to the length equal to 3/4 of the length of the kernel. That I told you, this is the ascospore. This should be 3/4 the length of the kernel. Then and only then we stop this germination. Then we consider the germination is over. Now the morphologically it looks like this. The dry barley grains look like this, and then after the steeping process the wetted barley grains looks like this, and after the germination of the barley grains it looks like this. This is the physical change how take place during the malting process. Now during drying, the germination is terminated by heat treatment. Drying is carried out thermostatically to avoid the inactivation of enzyme. We maintain the temperature in such a way the inactivation of enzyme doesn't take place, and application of the heat develops the color, flavor and aroma, and final moisture content is, I told you, 4 to 5% is desired. Then lower temperature for a light malt or vice versa. So if you increase the high temperature, then color will be little bit darker, low temperature color will be little bit lighter. Now this is the color, that different color, that malt that we have with the lager beer. This is the kind of pilsner, that pale ale, that is like this. And the dimmer, you can see we can see the color of the barley grains also varies with as per our as we change the characteristics of the barley grains, that color changes. So we have different grades we have. This chart gives the rough idea about the color contribution of varying amount of different malt type in beer recipe. So use of malt has the different use. Brewing industry as the cheap raw materials. Then it is used as a saccharifying agent in the manufacture of industrial alcohol, because I told you that whiskey is produced from the starchy raw materials, the wheat starch and rice starch. So there we can use this hydrolytic enzyme for the hydrolysis of the starchy material. So a saccharification agent in the manufacture of the industrial alcohol, it is largely used. Malt is used in the manufacture of malted milk. I told you that the malted food is usually recommended for the school children and for the patients, the candies, cereals. Then food coloring, it is largely used. Then next step is the mashing process. The purpose of mashing is to digest or to dissolve as much as possible of the valuable portion of the malt and malt adjunct. I told you that here the insoluble material will be solubilized. The media which is fermented to produce beer is called the oat. This is very important. The oat tank, if we go to the industry, we will find the oat tank. Oat tank is nothing but after the saccharification, whatever soluble material comes out, that is oat, because the media that is suitable for the fermentation purpose, that is called oat. The sweet oats that results from the enzyme process containing the dextrin, maltose and other sugar plenty protein degraded products, minerals, tannin and coloring matter and other substances. The meshing process, as I mentioned before also, it can be divided into two types. One is called infusion, another is called decoction. Now, infusion process, we have upward method and downward method. Basically, upward method means we increase the temperature gradually, that initially proteolytic enzyme, they will be activated, they will degrade the protein molecules. Then finally high temperature, 60 above 60 degrees centigrade temperature, that saccharification take place, where alpha amylase, they degrade the starchy molecule to glucose. And downward method is start from the high temperature to low temperature. Decoction process, basically the proteolysis take place in two different temperatures, and saccharification take place in two different temperatures. That is called the decoction process. Now, boiling of hops, that I mentioned in the last lecture, this is very important step as per beer making industry is concerned. So it is main purpose of boiling of hops is that oat is concentrated, that when you boil it, then the water evaporation will take place, that causes the things concentrated, and also the oat will be sterilized, that if there is any kind of contaminants present, that will be killed. Then it inactivates the enzymes, because with the enzymes is produced during this germination process or used in the during the massing process, that will be inactivated. Then it extracts the soluble substances from the hops, because I told you hops contains the resin, tannin and the essential oils that will be solubilized, and during this process then it precipitates the coagulable protein, because I told you the tannin and protein, higher protein molecule particularly, that precipitated out, and other solubles is slightly caramelize the sugar. I mentioned during the alcohol fermentation process how the caramelization takes place. When we heat the sugar, dehydration of the sugar takes place, and due to dehydration the color of the sugar changes to black. So slight caramelization means the black color development will take place. And then lager beer contains lesser quantity of hops, because the water found hops is liquid per barrels, and in case of alley beer it contains higher amount of hops, about 2 pounds beer per barrel of beer formation. Now, in the fermentation, the addition of yeast to the oat, you have to use for the carrying out the fermentation. Only pure culture is usually used in the fermentation. Saccharomyces cerevisiae commonly used. In case of lager alley beer, we use Saccharomyces cerevisiae. This is considered as the top fermenting yeast, and Saccharomyces convergensis is usually bottom fermenting yeast. Following changes take place during the fermentation of oats: conversion of most of the fermentable sugar to ethanol and carbon dioxide and glycerol and the acetic acid. Protein derivatives are converted to the into the higher alcohols. So this is the difference we have between the top fermenting yeast and the bottom fermenting yeast. I told you the top fermenting is Saccharomyces cerevisiae, bottom fermenting is the Saccharomyces convergensis, and they rises the surface. This is usually take place at the higher temperature, and time of fermentation will be less as compared to this Saccharomyces convergensis, which usually take place at the lower temperature. Now, the material of the fermentation, that is the materials of the fermentation tank. In ancient age, wood or concrete tanks are used, short life and contamination risks. So nowadays we use some carbon steel storage tank as the alternative. It coated with epoxy resin in order to check the corrosion. Epoxy resin largely used in the fermentation industry. It protects the acid attack to the material of construction. It has the following property: it can resist the repeated sterilizing temperature. It has the longer life, give no toxic product and good thermal conductivity and higher mechanical strength. These are the different properties that we have. The material of construction that we have with the fermentation broth, this is to be maintained. Now maturation process, I told you, I mentioned before, the beer is stored at the refrigerated cellar at 0 degree centigrade, and during this process the unstable protein, yeast and resins and other undesirable protein are precipitated out, which is very important. Then esters are produced to give the characteristic flavor. I told you the ethanol and organic acid, but they form the esters, so the harshness of the beer will go, and then harshness disappeared, what I mentioned. Then antioxidant like sulfide are added during the storage because they increase the storability of the particular beer. Beer also make peel poop by using special enzyme. I told you but during this maturation process we add some kind of proteolytic enzyme. Main purpose of adding proteolytic enzyme is to degrade the higher protein molecule into the smaller protein molecule, so that even in low temperature the protein will not be precipitated out. Now finishing operation, the matured beer is carbonated under pressure using the carbon dioxide that is at least 99.5% pure until final carbon dioxide content is 0.48 to 0.52% is obtained. This the function of carbon dioxide is several. It improves the quality of the beer because beer is a carbonated drinks, and I told you this protein helps for maintaining the foam in the beer. Anaerobic condition is maintained due to the replacement of oxygen. So is the anaerobic condition, a few organism can grow, the contamination problem will drastically reduce. Then helps in the preservation of beer because this anaerobic condition that increase the preservation of beer because less number of organism can aids in the production and retention of foam. Retention of foam is very important. I told you that carbon dioxide in presence of protein it form the foam which is quite stable. So you can remain for some time. Then carbonated beverage is cooled and filtered with diatomaceous earth, because here let me point out sometimes we find some kind of fine particles present in the beer, and this is removed by using some filter aid. The filter aid means I mentioned before also, this is largely used in the different chemical and biochemical industry. Particularly, I try to mention that in case of chemical and biochemical industry, we use the activated charcoal, which is very fine particles. When we use that, and then after this has the bleaching powder. This has the bleaching property. This bleaching property helps to remove the color from the product. So. But after use of activated charcoal, it is very difficult to remove the activated charcoal because size is very small. So what we use some kind of filter aid so that we can bigger particle can the activated charcoal can absorb on the surface of the bigger particle, and that bigger particle we can separate it out very easily. So that is here diatomaceous earth is kind of filter aid that is used in the beer making industry to separate the fine particles. Now I told you that racking process, that is another process that is this is also kind of through this racking, the kind of clarification process. Clarification, I mentioned that it is kind of separation of solid from the liquid. If you want to separate the finer particles, you have to pass through the racking process so that fine particles can be separated out. The beer bottle is pasteurized if it is not consumed for the short period. I told you the beer can be marketed in two different forms. One is short time market and another long time market. If you have short time market, you don't have to pasteurize. You use the preservative like normal heptyl parahydroxybenzoate you add, that increase the storability of the beer, so you can have a short time market, this can be used. But long time market, we do the pasteurization at 60 to 61 degrees centigrade for 20 minutes. And three type of pasteurization we have: fill, cool and heat. Fill, close and heat. Heat, fill and close. Heat, cool and fill. So first you fill, then close, then heat. This is that. And first you heat, then fill, then cool, then heat, cool, fill. So there are three different the mode of operation we have for the pasteurization of the beer. Now beer has several defects that we have. How defects of beer, and defects of beer is arises due to mainly due to the precipitation of the proteins or remaining some suspended particles. So this can arise due to the unstable protein tannate complex, starch, resin and the microorganism etc. Occurs at low temperature, occurs when molds has been improperly dry or in the kiln, and when a barley with very high protein content is used. That this can be removed by solution is warming the beer and the pasteurization process. So this can be removed to some extent, that the turbidity of the beer can be removed. But another very important defects of the beer is the faulty beer. This is due to the following reason: due to the use of low grade raw materials in the mass. If the quality of the raw materials is very less, very low grade, that particularly quality of the barley grains is the poor quality, then there is the possibility of these defects in beer. Then we have hops of poor quality. If the hops have very poor quality, that also gives some poor quality, poor defects in beer. Then too much of hops. If you use too much of hope, it gives too much of bitterness in the beer. And prolonged boiling with hope, if you heat the beer for longer period of time, then also it causes some kind of defects in the beer. Contact of the beer with iron causes the inky taste, because the iron if contains iron contents in the beer, that causes the inky taste, because the ink has some kind of taste. Particularly in the early days we use the fountain pen, and in the fountain pen we use the ink, and that ink if you taste in the tongue will have typical taste, and that is called the inky taste. And this inky taste is occurred due to the presence of iron in the beer. Then contact with tin, then also it is there, it is a problem. Then unsuitable for the brewing water if is used, that causes if the quality of the water is poor, then also causes the faulty in beer. Young and green beer, that is also due to the formation of that. Carbon dioxide deficiency also causes the defects in beer. So there are different defects in beer that we have. So then composition of beer depends on the different parameters. One is the maturity and quality of the raw materials, malting process, type of fermentation, maturation process and finishing operation. The types of beverages, we have alley beer prepared by the top fermenting is Saccharomyces cerevisiae. It is pale in color, and it is tart in taste, and it has high alcohol contents, and it has good hops. It has more hops. So that this is alley beer. That looks like this is a bit darker in color. That is not in practice in India, but some places in the world they use this dark color beer. But in India we mostly use the lager beer. Lager beer is little bit yellowish in color. You can see this is how it looks. It's stored beer, and it is prepared by bottom fermenting is Saccharomyces culvergensis. It contains the high alcohol and low proportion of hop, because I mentioned that quarter pound hops is required per barrel of the beer. And then we have the porter. porter is further, if you consider that, but the dark color beer, because it is very very dark. If you consider that alley beer, it is much darker than alley beer. You can see it. It is much darker. What we call stronger than alley and lager beer. That porter is stronger actually, and high in extract alcohol, sweet in taste. This is porter has sweet in taste. It is prepared from the dark malt. That is why it is dark color, and I mentioned that how the dark malt is taking place. If you barley grains, if you heat it, try to go if you char it, if you heat it for quite some time, then the color will be little bit darker. So we can easily make the color darker. They usually produce from dark malt. It has less distinct flavor than the alley. It is less distinct flavor than the alley. Now we have this is the difference between the alley and lager beer. 1,000-year-old relatively lager beer is a little bit new. And I told you that alley beer, the fermentation takes place a little bit higher temperature as compared to the lager beer. It is the alley is formed by the using top fermenting yeast, the bottom fermenting yeast. Alley is produced by using Saccharomyces cerevisiae, and lager is produced by using Saccharomyces culburgensis. Quick brief cycle. This is 7 days. It is a longer cycle, that is several months. And usually the brewed between 59 to 77 degree Fahrenheit, usually brewed between 40 to 45 degree Fahrenheit. Strong, assertive, and most robust in taste, the smoother, crisper, and most subtle in taste and aroma. Served not too cold, usually 50 to 55 degree, 10 to 14 degrees centigrade, sometimes called cellar temperature. Cellar, I want to tell, cellar is nothing but the underground tank that is used for storing the for the maturation of the alcoholic beverages. And larger, the lager is the served cold, usually 40 to 45 degree Fahrenheit, and that is equivalent to 4 to 7 degree centigrade. Now a malt beverage, we have another. So we have malt beverage stout. It is very stronger than porter. We have seen the porter is quite dark. It is more darker than that, and dark in color, high alcohol. It has strong flavor of malt and prepared from dark malt and in sweet in taste. This is called stout, and this is rock beer. It is very heavy beer. It is dark in color and high alcohol. So in conclusion, let me tell you that brewing industry that is largely used throughout the world, and we have two different type of beer largely used throughout the world. One is lager, another is alley beer. And barley grains is mostly used as a basic raw materials for the production of beer. But with barley grains, we use some kind of malt adjunct also. That four steps are major steps are involved for the beer making industry. One is malting, another mashing, and there is hopping, then you have fermentation. I told you the maturation process also very important as well. Beer making industry, long time and short time marketing. On the basis of that, you have to find out that whether beer should be pasteurized or not. And your malting process depends on different steps. So we have steeping process where your barley grains is wetted. Then you have to do the germination on the wet and aerobic conditions. And in case of In case of this then mashing process, you solubilize the insoluble material, and in the ferment, then hopping, you boil with the hops to get the desirable ingredients from the hops, good the desirable taste and flavor to the beer. And finally that we pass it through the fermentation process. Two type of yeast is used. One is Saccharomyces cerevisiae and Saccharomyces convergensis, and after that it passes through the maturation process, and we do the carbonization and do the bottling or put it in the can or the barrels and send it in the market. So this gives some kind of idea how the brewing industry looks. I think in the next class we will be discussing some other fermentation process. Thank you.